Okay. Okay. So if that is okay with you, I'm going to start. Stan?
Yes.
Okay.
Give me 1 second. I'm having trouble getting this to show. So I think what you're going to see is you're not going to see the slide show, but you're going to see. 1 second.
Yeah, don't worry. So, vamos a esperar un momento. Otra vez, buenos días a todos. Están entrando más al salón. Qué bueno, me da gusto. Y en cuanto el doctor Stan me diga que estamos listos, lo iniciamos el seminario. Les recuerdo las indicaciones: mantener micrófonos apagados y pantallas apagadas. Are you okay, Stan?
I'm ready.
Okay, so I'm going to introduce you in Spanish and then I'll let you continue, okay? Okay.
Perfecto.
Bueno, muy buenos días a todos de nuevo. Es para mí un privilegio presentarles al doctor Stanley Maloy, quien es un destacado genetista en los Estados Unidos. Al doctor Stan Maloy tuve la fortuna de conocerlo cuando estaba haciendo el doctorado en el IBT. Hace ya un buen número de años, estábamos platicando, en el 2002, 2001. Y desde entonces me he mantenido en contacto con él. Es una persona muy amable, es muy interesante. Y me voy a permitir leerles algo de lo que... ¿Podrían, por favor, apagar su micrófono? Es muy importante que apaguen sus micrófonos, por favor. Por favor. Ok, ya lo saqué. Así le voy a hacer. Lo lamento mucho, pero si alguien no tiene cerrado su micrófono, lo voy a botar del salón. Tengo el poder de hacer eso y no lo voy a dejar entrar. No, lo lamento mucho, pero esto es algo serio. Bueno, me permitiré presentar de nuevo al doctor. El doctor Stanley Maloy es vicepresidente asociado de investigación e innovación y profesor de microbiología en la Universidad Estatal de San Diego. Obtuvo un doctorado en biología molecular y bioquímica de la Universidad de California en Irvine. Realizó una investigación postdoctoral en genética en la Universidad de Utah. Luego, fue profesor de microbiología en la Universidad de Illinois Urbana-Champaign durante muchos años. Ha realizado cursos y conferencias en todo el mundo y ha recibido numerosos premios de enseñanza. Es miembro de la Academia Estadounidense de Microbiología. Recibió múltiples cátedras honorarias, un doctorado honorario. Fue ya presidente de la afamada Sociedad Americana de Microbiología, la ASM. Ha asesorado a agencias estadounidenses e internacionales en investigación, bioseguridad, biodefensa y salud. Trabajó con empresas agrícolas, farmacéuticas y biotecnológicas. La investigación de Stanley se centra en cómo surgen nuevas enfermedades infecciosas. También ha sido coautor de más de 100 publicaciones y de 10 libros, uno de los cuales es el libro que nosotros usamos en nuestro curso, llamado Freyfelder, el cual ha salvado a varios estudiantes y a varios profesores para cuando damos los temas. Además de que ha creado diversos programas educacionales, así como sitios de internet. Ha hecho diversos videos educativos referentes a la microbiología, organiza cursos y conferencias en todo el mundo. A mí me hubiera gustado tener a Stanley en vivo en la escuela. Malamente no conseguí recursos y en estos tiempos ya no se puede. Entonces, para mí es un privilegio. For me, it's just an honor, Stan, that you accepted to give this talk. I'm really, really happy that we were able to finally see each other, at least through a camera. But it's just my pleasure. Thank you.
Muchas gracias. So first, I apologize that I am going to speak in English. I have spent enough time in Mexico and South America and Spain and Cuba that I should speak Spanish well, but I don't speak well enough to give a seminar. So I have translated my slides, and then I'll speak in English. So my slides are in Spanish, but I'll speak in English. So hopefully that will help make sure that we're all on board all the time. And I also, when I'm speaking in a different language, tend to speak relatively slowly, so it shouldn't be a problem. I should add that if it's okay with Antonio, I'm happy with answering questions in between if something comes up. So just let me know. So today I'm going to tell you about how genetics can really serve as a functionator. We talk so much about functional genomics, but genetics is really a way of understanding functions. And I'm going to talk about how we use genetics to understand host specificity in Salmonella. This involves a lot of people from my lab, but it also involves a very long time collaboration of Dr. Guido Mora from Chile. Guido is the one who convinced me to begin working on Salmonella typhi, and we have collaborated for a long, long time. So when you think about host specificity, one of the real key reasons this is important is because of its role in thinking about emerging infectious diseases. And I like this cartoon. It fits so well with our current situation with coronavirus. It says that thus the end of the world is not going to be from an explosion or a bomb. It's going to be from some new infection. And it really defines the situation that we're in right now. So when you talk about emerging infections, you can say, where do the new infections come from? Right? And the answer is really two types of things. Almost all new infections in humans come from animals. About 75 to 80% of infections in humans that we haven't seen before come to us from animals. The other thing that happens is during adaptation to new niches, then these new diseases can acquire different properties that allow them to be more infectious. Right? So if you ask, how do the new diseases evolve? There's two main ways that they evolve. One is by acquiring new genes from the environment. An example that my lab works on a lot has to do with acquiring exotoxins, bacterial exotoxins from the environment. But the other way this can happen is by mutations that change the genomic makeup. An example of that is host specificity that I'll talk about today. And if I have some time at the end, I'll talk a little bit about the One Health concept and how that ties into everything that we're talking about today. So the main character we'll talk about today is Salmonella enterica. And there are over 2,500 serotypes of Salmonella enterica that can cause disease in humans. If you look at all those serotypes, they fall into 3 different categories with respect to host specificity. There are the generalists. This includes Salmonella typhimurium, Salmonella enteritidis. They can infect virtually everything that can walk, crawl, or fly on the face of the earth. And now I have to say some things that can't walk, crawl, or fly because they also can infect plants. There are host-adapted Salmonella. Salmonella Dublin is adapted to cattle. Salmonella choleraeus is adapted to pigs. These organisms are retained in that population and maintained in that population. They can cause disease in other organisms, but as a rule, it doesn't linger in the other organisms. And then there are host-specific Salmonella. Salmonella typhi causes typhoid fever in humans. It doesn't even cause a serious disease in great apes that are very closely related to humans. It's human-specific. Likewise, Salmonella pylori causes disease in chickens, but it doesn't cause disease in other organisms. The test that we have for host specificity is a biopsy mouse. So if you do an infection of Salmonella in a biopsy mice and you inject interperitoneally, typhimurium has an LD50 of less than 10. Probably a single bacterium can kill that mouse. In contrast with Salmonella typhi, the LD50 is greater than 10 million. There's a certain point where LD50 doesn't count anymore because the amount of lipopolysaccharide associated with the bacteria would kill the mouse even if this bacterium is dead, right? So this is over a million-fold difference in sensitivity, right? So we were interested in the question, how can you figure out which genes in the bacteria determine host specificity? Which genes distinguish typhimurium and typhi to allow this infection in mice? So if you think about evolution of host specificity, you can think about several kinds of genetic changes that might happen. One change might be that you could acquire genes, like a genetic island, that would give you a gain of function. This might also happen from missense mutations. A second thing that might happen is you could have a mutation that alters the function, a missense mutation. And a third thing that could happen is you could have a deletion, a nonsense mutation, a point mutation, some mutation that results in loss of function. And it's that loss of function that's really important in this process. Any of these possibilities could be true. To try to figure out which one of those might be important, very early on, we began to do genome sequencing and comparative genomics. We did our work back when there were very few Salmonella strains that had been sequenced. But now there are over 900 Salmonella strains sequenced, 900 sequenced, and the story is still basically the same. If you compare any two Salmonella, they're about 97% identical in their common genes. They share very common virulence loci with a few exceptions. About 10% of the genome of every different Salmonella is unique. And the host-specific serovars have about 4 times more pseudogenes than the generalists. The other thing that is noticed is that the host-specific serovars have rearrangements in the ribosomal RNA, RRN operons. A lot of this genomics was done, but in the end, it was impossible to know which ones of the changes are important. And we had genome sequences, there were lots and lots of changes, but how do you know which are important? Right? So to answer this question, we thought we would use 3 genetic approaches. The first approach was, we thought there was a possibility that typhimurium, Salmonella typhimurium, has a murium gene that allows infection in mice. It could be a gene, it could be multiple genes. And so taking a gene library from Salmonella typhimurium and bringing it into mice would allow those mice to cause infection would allow those hybrids to cause infection in mice. Okay? So we made cosmid clones that represented the entire genome, and none of those cosmid clones from typhimurium would allow Salmonella typhi to infect mice. Secondly, we thought, well, maybe Salmonella typhi has a gene that prevents survival in mice. And if that was true, we should be able to get a mutation that disrupted that gene. So we tried a lot of approaches to find mutants that allowed typhi to grow in mice. We never obtained those mutants. Right? Now, there was a good indication that these 2 approaches weren't going to work anyway, because 2 really well-known, distinguished Salmonella experts, Stan Falco and Roy Curtis III, had both tried these approaches and both of them failed. Right? So we thought, well, if these aren't right, maybe the issue is that we need both to bring in something from typhimurium and to eliminate something from typhi. The solution to this was to use recombination, where we made hybrids that brought typhimurium genes into typhi, so genetic hybrids. And this approach had never been tried before. So this is the chromosome of Salmonella, and the chromosome is divided up into 7 of these RRN genes, and they're labeled. So they go from A, B, C, D, E, F, and G. So we made derivatives where we brought in a region that allowed transfer of the chromosome right from here and allowed it to go all the way to here. So we substituted that big region of the chromosome, or we substituted this big region of the chromosome, or this region, and so on. So we made large substitutions that brought in typhimurium genes into Salmonella typhi to allow this to happen. By the way, this sounds really easy. It was a lot of genetic trickery to be able to do this experiment. In the end, what we found was if you brought in any single region, it could have a small impact on growth in mice, but it didn't allow full growth in mice. And any 2 regions allowed a little bit more growth in mice, but didn't allow full growth in mice. So the conclusion of all of this work was that there must be multiple different loci around the chromosomes that are important, but it's an additive effect. So each one has a small impact. Right? So we set about mapping those loci that had a small impact. And what we noticed was that whenever we mapped one of these loci, very often it was tied to a place in the genome sequence where there was a pseudogene. So this region right here seemed to have a small impact, and it was a pseudogene in Salmonella typhi and wild type in Salmonella typhimurium. This gene, this region right here, carries the SOP2 gene, which is wild type in Salmonella typhimurium and a pseudogene in Salmonella typhi. Right? So this led us to wonder, are these pseudogenes important for host specificity? Now, this is a tricky thing to ask because you have to be able to substitute a very small region of the chromosome precisely. And so to do this, what we wanted to do is the first thing we did was we interrupted a gene in Salmonella typhimurium. And we did that by PCR of a locus that had chloramphenicol resistance and tetracycline resistance, plus a little bit of that host region at the ends of the PCR fragment. So this produced a PCR fragment like this. These are antibiotic resistance in the middle, and this is the host genome at the end. We could then recombine that into Salmonella typhimurium by selecting for the chloramphenicol resistance, and it will precisely substitute a very, very small region of the Salmonella typhimurium chromosome. Then what we did was we took that same region from Salmonella typhi, we did PCR of the Salmonella typhi chromosome. We then took this PCR fragment and we substituted it in to the typhimurium chromosome that we had just constructed. And we could do this by selecting for sensitivity to tetracycline, and allows us to very precisely exchange a very small region between the 2 chromosomes. So if we want to substitute a specific pseudogene, we can look at that pseudogene without worrying about what's happening with adjacent regions of the chromosome. Okay? The key trick to this is having a selection for insertion, chloramphenicol resistance, and a selection for loss of that fragment, and that's tetracycline sensitivity. Okay? When we do this, we could then go in and sequence the chromosome. And this just shows this is a Salmonella typhi wild type strain, and it's got a pseudogene, a loss of a base here. Here's the typhimurium chromosome. It's got a wild type SOP-D gene. It's got a C at that position that's missing in Salmonella typhi. And these are these regions right here are hybrids. So these have a typhimurium chromosome that carries the typhi pseudogene. Now we sequenced way out from here and we showed there were no other changes in this gene. Right? We also could show that those changes, making these genetic hybrids, bringing the one pseudogene from typhi into typhimurium, did not affect growth of those hybrids in vitro on a petri dish or in lab medium. So the next question that we wanted to ask was, do those particular pseudogenes have an impact on the virulence, the host specificity in mice? So in this case, we did infections. We injected interperitoneally into BALB/C mice that are very, very sensitive to Salmonella. We began with a ratio 1-to-1 of our mutant, our hybrid strain, to the wild type Salmonella typhimurium or Salmonella typhi. And when we're done, we look after the infection process occurs. We ask, when we take the bacteria out of the mouse, we say, what is the ratio of mutant to wild type? If that ratio is 1, that mutation has no effect on virulence. If, however, the amount of mutant is much less than wild type, it indicates that that mutation decreases virulence relative to the wild type. Okay? So this is a simple competition assay using mice as a host. If we look at the results of this competition assay, now we're comparing typhimurium versus typhimurium strain that carries a Salmonella typhi pseudogene. The gene is SOP-D, so we look at our derivative that we made that has a SOP-D knockout mutation, and that has a decrease in competition. It's about half as effective as the wild type. We look at a SOP-D with our pseudogene inserted, and it decreases competition to the same amount. We could also do this with the SSEJ gene, and we see the same effect. The full knockout with the insertions decreases partially the competition, and the pseudogene decreases to the same amount. These results tell us that the pseudogene has as big of an impact as a complete knockout mutation, and indicates that that pseudogene is really a knockout mutation. It also indicates that that pseudogene has a small effect on competition, not a complete effect. Likewise with this other pseudogene. Now we construct a derivative that has those two pseudogenes together, and we can see it has a much greater impact on competition in the mouse. So the results here suggest that the decrease in virulence in mice from Salmonella typhi is associated with multiple mutations, not a single mutation. There are a lot of other Salmonella typhi pseudogenes that have an effect on growth in the mice. For example, a lot of pseudogenes that affect vitamin B12 biosynthesis or metabolic pathways that depend on vitamin B12 are wild type in Salmonella and a pseudogene in Salmonella, wild type in Salmonella typhimurium, and a pseudogene in Salmonella typhi. The work of Andreas Baumler's lab shows that these particular genes are really important for Salmonella typhimurium to live in the intestine. And since Salmonella typhi causes a systemic infection, it doesn't survive long in the intestine, these genes are not important for infections by Salmonella typhi. The key take-home point is that pseudogenes can have a big impact. Loss of function genes can have a big impact on infection of Salmonella. So again, the take-home points are that Salmonella typhi has more pseudogenes than Salmonella typhimurium. Those pseudogenes are null mutations. Those pseudogenes limit the growth, the systemic growth of Salmonella typhi in mice. And those pseudogenes limit the growth of Salmonella typhi in the intestine. So the conclusions are that the accumulation of multiple pseudogenes restricts certain niches in the host. And because those pseudogenes map at many different places on the chromosome, it explains why we were unable to find derivatives that influenced one particular part of the chromosome that allowed growth in the mouse. All right, now when I was a graduate student, we were told that pseudogenes happen in eukaryotes, but they're extremely rare in bacteria because bacteria grow very fast. And so bacteria would not keep around genetic material that wasn't important. They would delete it. So does this increase of pseudogenes in the host-specific Salmonella, is that increase due to a decrease in intermolecular recombination? So one way you can test this is you can look at recombination between inverted repeats that result in inversions. And this can be done very easily by mapping ribosomal RNAs and doing pulse field gel electrophoresis. It was done by Liu and Sanderson, and they showed that in fact, there were a lot of these kinds of inversions that occur in Salmonella typhi. So if you look at Salmonella typhimurium versus Salmonella typhi, and you look at the RRN pattern, this is the origin of replication, and this is the terminus. If you look in Salmonella typhimurium, you can see that they go D, E, F, G, A, whereas in Salmonella typhi, these genes have undergone rearrangement so that now it's E instead of D, F, D, B, A. So there have been a lot of changes in Salmonella typhi, and we've looked at Salmonella typhi from around the world, as have Liu and Sanderson. And in that case, we find these rearrangements happen very often in Salmonella typhi, and we always see typhimurium with the exact same arrangement. Okay? So these rearrangements between the generalist bacterium and the host-specific bacterium were another big change that occurred. The fact that they occur so much in typhi suggests that those pseudogenes are not there because of a loss of recombination. They're there because they play an important role. So if you say, all right, how come the genomes of the host-specific Salmonella are so plastic? How come they undergo these rearrangements so much? You could make two predictions. You could say, well, this is a biochemical difference between typhi and typhimurium, or it's an ecological difference between typhi and typhimurium. So a biochemical difference would mean that there was a mechanistic reason for this event, and an ecological difference would mean that the event might be able to happen in both organisms, but there's a selection that happens in nature so that we see the events in typhi, but not in typhimurium, right? So the test for these two hypotheses is to measure the frequency of those rearrangements in typhi and typhimurium. Okay, so the predictions are if there's a biochemical difference, the frequency will be this, the frequency of recombination will be the same in both. And if there's an ecological difference, then we will observe many fewer in vitro than in vivo in Salmonella typhi. Let me restate this. If it's a biochemical difference, that difference will be observed in vitro when growing in the laboratory, as well as when growing in a host. If it's ecological, we expect that the frequency will be lower in the laboratory than observed in the host. Now, we can measure those differences using PCR, but to measure a very low frequency event like this by PCR, it just wasn't sensitive enough. People may find that very surprising because they think of PCR as being very, very sensitive. But there are times PCR just isn't sensitive enough. So we developed a genetic test to look for inversions at the RRN operons. So what I show you in this figure is these boxes are ribosomal RNA operons. And what we did was, at the very end of one ribosomal RNA, we inserted a chloramphenicol resistance cassette. And at the other ribosomal RNA, we inserted a kanamycin resistance cassette. Okay? So if this particular derivative does not undergo any change, and we grow bacteriophage on it, that phage can package up chloramphenicol resistance from here, or kanamycin resistance from here, but you'll never see a particle that carries both chloramphenicol and kanamycin resistance. They're just too far apart. However, if this derivative undergoes the recombination event, so it starts here, it recombines here, it goes down, it comes up, it recombines down and back. Now what we get is one RRN that's got both chloramphenicol resistance and kanamycin resistance, and another RRN that doesn't have either. So if we grow phage on this derivative, some of the particles will carry both of these antibiotic resistance molecules. So by simply using these phage particles to transduce into different hosts, we can answer the question, have they undergone recombination or not? It's a very, very, very sensitive test that gives us sensitivity at the level of something on the order of 10 to the minus 12 to 10 to the minus 14, as sensitive as we can possibly measure. And now once we've done that, we can ask, what is the frequency of inversion? So we looked at the number of inversions by this assay. We confirmed them all by PCR versus the number of colonies that we selected, okay? So that is the number that inherit one antibiotic versus down here versus the number that inherit two antibiotics. In Salmonella typhi, two different strains of typhi and Salmonella typhimurium, we saw that the frequency of inversion was the same in all cases. So this answers a really important question for thinking about that biochemical versus ecological process. Okay? The frequency that we observed in vitro was the same for both typhi and typhimurium. And it indicates that the difference is not a biochemical or physical difference between these two different bacteria, but in fact, it may be a difference in their ecology, in their lifestyle when they infect a host. So if you say, why might these two have different ecology? We can compare generalist and host-specific Salmonella. Generalists have an acute infection. Host-specific have a chronic infection. Generalists have real high levels of competition in nature. Host-specific have very limited levels of competition in nature because they have a very limited host range. The population of generalists can be very large because they can be in so many different places in the environment, but the host-specific bacteria have a very small population because they can only infect people. And though we think there's a lot of people on Earth, it's very, very small number compared to the number of mice and cockroaches and other organisms. The generalists have environmental reservoirs. The host-specific bacteria have no known environmental reservoir other than humans. And the generalists don't very often undergo a genetic bottleneck because their population is so large. In contrast, the host-specific bacteria go through a bottleneck very often. An infection will go in a population and a single person may transfer that infection to a new population. So every time that happens, they undergo a bottleneck. Okay? So the prediction of this is that the ecological difference between these bacteria most likely occurs within a host. Now, looking for differences during infection in humans is not a very easy thing to do for Salmonella typhi, because Salmonella typhi causes very long-term infections, these chronic infections, and having access to people over a long period of time with the chronic infections is not very easy. However, we're very lucky to collaborate with a group from the Robert Koch Institute in Germany. This Robert Koch Institute is in what was formerly East Germany, and that institute, for a long time in East Germany, there were problems with people infected with typhoid fever, but they didn't have a very good supply of antibiotics at that time. So when somebody had typhoid fever, they would take a stool specimen from them, isolate the Salmonella typhi, and then they would store it. And then what they would do is they'd tell the person to come back. And the person came back, and a year later they tested, and if they still had Salmonella typhi, they stored it. And so they had these long-term samples that came from people who had chronic infection for decades. Not a very good way to treat patients, but a really good sample set for our experiments. So we wanted to know if these patients, these chronic patients, very often were asymptomatic. So we had these chronic patients that we had taken they had taken samples over a period of 22 years, right? We compared the samples based upon a genetic process called phagotyping and the organization of the chromosome based upon rearrangements between RRNs. The phage typing of every organism that went in was the exact same as when it came out. So we know that we're looking at the same organism, not reinfection of these individuals. So then what we did was we took samples. And now, because we had a much smaller sample set, we used PCR to look for rearrangements. And here's the basic idea. So these are two sample RRNs in the chromosome. If we take this primer and this primer, those two primers will work to PCR up the RRN G fragment, so that's plus. If we take P3 and primer 4, they will PCR up our RNH, so that's plus. But if we take P1 and P4, they cannot make a PCR product. They're too far away from each other. Okay? And in fact, they're pointing in the same direction. Likewise, P2 and P3 won't produce a fragment. But if a recombination event occurs that causes an inversion between these fragments, now we have a hybrid derivative. And in this case, primer 1 and primer 4 will produce a product. Primer 2 and primer 3 will produce a product. But primer 1 and primer 2, worked up here, will no longer produce a product. So we could do these PCR tests on lots of samples. And the question is, between a sample that was put away 1 year and another year, do we see these kinds of rearrangements? This just shows an example of one patient. This is patient number 4, right? And you can see, if you look at the rearrangement type in this patient, you can see that in 1976, their Salmonella typhi had 2 arrangements. By 1981, only one of those arrangements had survived, and it was the same typhi in 1983. By 1986, some new derivatives had occurred, and one of those ended up being predominant in 1987, and it stayed the same until 89, and in 89, a whole new set occurred. So over these periods of time, rearrangements occur at some low frequency, and once they've rearranged, one particular strain may predominate. We did this with each one of these different samples, and what we saw was that rearrangements happened in the human host, but when we look at samples that had been stored in a test tube, they didn't change. A sample that had been stored away in 1976 was the exact same in 1991, right? So this tells us that it does not, these rearrangements do not happen in a test tube, but they happen during the infection of humans. There's something about growth in humans that allows them to undergo these kinds of rearrangements and actually provides a selection for those rearrangements. So here is how we interpret these results. If there's a new niche, for example, this chronic infection in a host, it provides new selection opportunities. Those new selections allow spontaneous mutations to occur and rearrangements to occur. When the strain passes through a bottleneck, what that does is it acts as what's called Mueller's ratchet. It selects for certain kinds of bacteria, and even if a mutation is non-important or has a very small impact, that mutation may be moved into a new host and may be saved in the population. And so what happens is, after you've accumulated enough of these mutations, like pseudogenes, it restricts the bacterium to a single kind of host. So in this case, it may be able to infect a human, but not a mouse. So if you look at generalist, host-adapted, host-specific, and obligate intracellular bacteria, this is an increase in specificity. If you look at pseudogenes, they happen in generalist. There's more in host-adapted, even more in host-specific, and a lot in obligate intracellular bacteria. If you look at rearrangements, they happen in generalists, similar frequency in host-adapted, a lot more common in host-specific, and they don't happen in obligate intracellular bacteria, because obligate intracellular bacteria undergo genome reductions and lose recombination functions. Likewise, recombination's functional in generalist. It's the same in all of these bacteria, but it's lost in the obligate intracellular bacteria. Right? So in this case, a real take-home point is that these host-specific changes, pseudogenes, and the rearrangements that happen in host-specific bacteria happen because of the selection that occurs within a host, not because of a loss of combination functions. So this was a way of going from genomics to genetics to study some functions. My lab and Guido Amora's lab looked at a lot of different pseudogenes and showed this to be a very consistent result. Likewise, some other labs, Andreas Baumler's lab has done some very nice work, basically did similar studies, and they showed other loss of function genes are important as well. And there are some gain of function genes that are really important too. So there's also been some other follow work that both supports and extends the results that I showed you above. So if you're interested in knowing some more about this, in this book that we wrote a while back called Microbes in Evolution, the World That Darwin Never Saw, it includes some stories about different ways that microbes undergo these kinds of adaptations. I told you a story about pathogenesis, but there are very similar stories in terms of thinking about plant pathogens or survival in certain niches in the environment and so on. And a really important aspect about this is thinking about One Health. And so that's a story I could tell as well. It's a short story and it relates to thinking about emerging infectious diseases and how they move from animals to humans and how we can best be prepared for the next pandemic that comes along. But right now, I've talked for about 50 minutes. And so I think it would be a really good time to stop and answer any questions that you might have.
Perfect. That's very interesting, everything you showed. So if you guys have any questions, please, is the moment. You can write them down and I can translate. Si tienen preguntas, por favor, adelante y yo puedo traducirlas. Y si no, adelante, abran su micrófono y ustedes pregunten.
Excuse me, Dr. Maloy. I got two questions about the host. The one is, what do you think about what can depend that a host is a specific host for Salmonella typhi or Typhimurium?
So I think that this is one of those accidents of evolution, right? So Salmonella typhi, by becoming a chronic human pathogen, it has a niche that is unique, right? So it's not competing with other bacteria. When it causes the chronic infections in humans, very often it lives in a biofilm on the gallbladder. And that's where it stays for a very long period of time. So it's a very slow growth phenomenon, but it allows survival of the bacteria. And it doesn't have to compete with Salmonella or E. coli or the other bacteria that live inside the gut of humans, as well as many, many other animals. So it restricts the Salmonella typhi in comparison with Salmonella typhimurium. Now, Salmonella typhimurium has to have lots of gene functions available that are lost in typhi, and those gene functions in Salmonella typhimurium play really important roles in allowing Salmonella typhimurium to actually invade, survive, and produce disease inside of the gut of different animals. Did that answer your question?
Yes, doctor, thank you. And I got another one, just to take my one note. Yes, if the reorganization on the host could activate some gene of resistance or virulent genes that are inactable, just for example, in the case of the kanamycin resist, and this reorganization can activate the express of these genes that are inactivate?
Right. So the rearrangements that we were looking at are between ribosomal RNA operons, and those operons are very big, and so they're about 7,000 bases, and they're very similar to each other at the different sites on the chromosome. So when the inversions occur, it just makes a hybrid ribosomal RNA. It does not make some derivative that will express new downstream genes. It just moves the material of the chromosome between those two sites back and forth, doesn't turn other things on, doesn't turn other things off, with the exception that in the very biggest part of the chromosome, it will move some genes a different distance from the origin of replication. And so that will affect the copy number of those genes during fast growth, okay? But you have to remember that in that chronic infection, Salmonella is growing very, very slowly. It doesn't have to grow fast. Growing slowly is a way that it can help evade the immune system and survive.
Okay, thanks, doctor. Thanks, doctor.
Muchas gracias, Rodrigo. Thanks, Rodrigo. So, anybody else? More questions, guys? ¿Más preguntas? Reitero, las pueden mandar por el chat o las puedo traducir yo.
I do have a question, Dr.
Go ahead.
Maloy. Can you hear me?
Yes, gracias.
My question is regarding the pseudogene insertion method that you used. Is there any way that I can find out more about it? Or how can I, get more detailed information regarding that?
Absolutely. Send me an email and I will send you information.
I very much appreciate that.
Yeah, in regards with that, thank you, Arturo. I have a question about that pseudogene. It was very, very clever the way you guys did it. I mean, you guys are geneticists, so you have the tools and the knowledge. So, how many of those recombinations, these pseudogene exchanges and things you guys did?
So we actually did. So my lab did some, and Guido Mora's lab did some. And so we did something on the order of 8, and the results looked pretty similar in all of our cases. And so we actually moved on and started studying some other aspects of this at that point in time. And some of those were there were some other groups that were working on a few other pseudogenes, and so we worked on the ones that other people weren't working on. It's a big enough question that we thought different groups could work together. The real important thing is that all of us found very similar results relative to the importance of pseudogenes. So,
yeah. So somebody else has more questions?
Can I ask you? Let me ask you all a question, all right? One of the things I told you was that we needed a genetic approach to detect the inversions because PCR wasn't sensitive enough. Right? Nobody asked me why wasn't PCR sensitive enough. We all know PCR is so sensitive, right? So here's the problem. In a bacterium, if you're doing PCR with this whole bacterium and you're doing PCR of a particular ribosomal RNA, once you've got some PCR products, they'll have a little bit, let's just pick this one, a little bit at the end and a little bit at this end, but they're going to have 7,000 base pairs that is homologous to this other RRN, right? So once you begin the PCR, now you're going to start doing aberrant PCR at all these other places, right? So it was a surprise to us what this problem was like, but it really limited our specificity. So we could detect an inversion that happened at 1 in 100 using PCR, and we could detect an inversion that happened at 1 in 10 to the 12th using our genetic approach, right? So it's a real interesting comparison of molecular and genetic approaches and when one might work and another might not. Both approaches are really important. Sometimes one of them has a real strong advantage over the other.
Yeah, that makes sense about the PCR, definitely. So more questions, guys? Questions?
I want to say something.
Oh, yes?
Yeah.
Yes, can you see this? Okay.
I can.
This?
Yes.
Dear Dr. Maloy, it's a pleasure to see and hear you through this platform. This is a particular experience to meet the author of one of our favorite textbooks in the microbial genetics course when we were young. That is yesterday. Thank you, Dr. Maloy.
Gracias. It's been slow coming, but I have a new edition of that book coming out. Kelly Hughes and I are writing, and so I really like to write books, and I love to hear people who use them.
Yeah. Okay. So. Thank you.
We definitely do, Stan. It's one of the working horses we have in the lab. The other one is the one I use is the I forgot.
No, nowadays we are using this. Yeah. This is the actual that we use in the course with the students. Yes.
Yes.
Neither.
But,
the last name.
But this is a classical text. Then we have to. Some days in some occasions we look for some concepts.
Yeah. Yes, I didn't update this book because the others were doing fine updating it. And so I moved on to other things at that time. There is a real difference in terms of emphasis of our two books. So like I said, we're updating it right now. One of the things that happens in the world nowadays is that many people who understand genomics don't understand why genetics is still important.
Yes. Right? That's very important. Genetics is very important, yes.
Right. The two go hand in hand and come together. This is, by the way, the hardest thing about a Zoom meeting is I love to talk with my hands, and the window is so small you can't do it. So if there aren't some questions, would you like to see a little 3-minute piece of fun?
Sure.
How many of you have seen the movie Contagion?
I did.
All right.
Guys, so you are alive, guys.
I actually haven't.
Okay.
I did it.
That's mandatory for a QBP.
Me too.
I saw it too.
I saw.
Great.
Okay. So if you saw it, some people who watched it looked at the movie, and then when the end of the movie and the trailers came on, they got up and walked out of the theater. And I stayed behind. And at the very end, so what this Contagion is about, it's a fiction story about a new virus, an emerging virus that comes about from Malaysia. This story, though, is based upon the truth. So it's based upon a new virus that had appeared in Malaysia, and people were very afraid of it causing a widespread pandemic, and it never did. But in the movie Contagion, it causes a widespread pandemic. At the very end of the movie, it shows a little spiel about how the virus that this movie was based on emerged, okay? And so it so happens that a friend of mine was a scientific advisor for this movie. And so he gave me a video clip of that last 2 to 3 minutes of the movie. And so we're gonna watch it now. It begins with a very dark screen because it's gonna be the jungle at night and there's bats flying, all right? Here we go. There's the bats. These are fruit bats. Of course, they like fruit. In this area of Malaysia, they really wanted to generate a lot of meat, especially for the growing tourist industry. So
Sorry, Stan?
where they grow these pigs,
Stan?
where they grow these pigs, yes.
I have a frozen image. I'm not sure if everybody else is seeing the video.
Oh, all
I
right.
just can see the,
Okay.
the title in section. It
Yeah, that's the only thing we can see.
Oh, you're not seeing the movie?
No.
Oh, no,
All
no,
right.
no, we,
Okay.
no.
All
You,
right.
you need to repeat that, please.
You know what? I think it probably takes up too much bandwidth. So
So, oops,
maybe my experiment won't work.
oops. Maybe
I'm really sorry.
Maybe you can share only, you can unshare the one that you have right now, and then share the one with the video. It shouldn't be that bad. I mean,
What I will do, Antonio, is I will share this video with you as
Okay.
long as you don't post it anywhere. Okay?
okay, that's okay. I'll do that. I'm guessing there's some legal issues that why it can be shared. But yeah,
Yes.
sure, I can do that. And what we can do, it would be great to have it here. Anyway,
I think it's a great conversation piece, especially given where we are right now with COVID and thinking about the future of infectious disease. So I'll send that to you and you can share it with the group. It's really very fun and an amazing case where a science story is told so well in a movie.
yeah, in my opinion, Contagion is one of the best told stories, because you remember the one with Dustin Hoffman? That was very exaggerated. That was really, really exaggerated. So this one is, let me just, so that one, it was really bad movie. I mean, it was funny, but this Contagion is really cool because it tells you the whole process when it starts, when they try to develop. Of course, they put some drama and some love issues and things like that, but I mean, actually, it's really, really cool, that movie. So if you guys haven't seen that movie, you should. It's like mandatory for chemicals, bacteriólogos, and parasitólogos. That's the career we teach here. And should be, because it's an amazing movie. And yeah. Sorry, Stan?
Yeah, okay. So, yeah, so I'm happy to answer any other questions that you might have. And if you don't have more questions, thanks so much. I really like talking to you. It's not near as good as being there in person, but it's better than not being there.
Yeah, you bet. I mean, you guys tienen más preguntas, yo la puedo traducir. Yo tengo más preguntas, pero me gustaría escuchar de los estudiantes, por favor. One of the other questions I have, well, it's more a comment. I mean, it's good that you mentioned that bacterial genomes and all genomes, even ours, are plastic. They're not like a written book that is written like the two tables, the two commandments tables that are written and that's it. No, they change. They change. They move. They recombine and all that. So it's your examples. Your example was really, really good, I think, in my opinion. So do you think that, now going to the question, is there a different kind of regulation in recombination for these bacteria when they do it? That's one question. And the other one is, have you guys looked for maybe other bacteria like Salmonella, like let's say Brucella that does this, or maybe another one that you can work in the lab? Staphylococcus, for example. I don't know, a different phyla of bacteria.
So we haven't. We have worked with different Salmonella though. So we've done a lot of work with Salmonella that are specific for chickens. So that's Salmonella polorum and Salmonella gallinarum. And we chose that specific model because, again, we have a host that we know we can work with, as well as we could use mice as the alternative host. And so basically with Salmonella polorum and gallinarum, it's a very, very similar story to this right here. We know from other people's work that in host-specific organisms, pseudogenes are in fact very common. So we expect that this is true in other cases, although often it's only a phenomenon that's recognized because of genome sequencing. It's never been directly tested.
Okay. So there is nothing like regulation in that part, like it's given some, I don't know, some environment signals that the bacteria eventually does a recombination. Nobody knows that yet.
Right. So at a certain point, when we thought we had many of the answers that we set out to figure out here, in this case, my lab has shifted, and my lab, the major thing that we are doing right now has to do with where new virulence genes come from in nature to generate new kinds of pathogens. And the answer to that is that most of them that we've detected come from bacteriophage in nature, and that many of the genes that we thought were specific for human virulence exist out there in nature in many, many different places. And they were probably selected for because they provide a resistance to protozoa that are a major predator of bacteria out in nature. Right? So it's an extension of the story trying to understand evolution of infectious disease, but it's taken us into many other organisms, right? A whole variety of other organisms, pseudomonads, and many, many different things. So.
Oh, that's great. I also like the, ¿alguien tiene preguntas, por favor? I also like the fact that you used some of the genetic tools that we try to teach here to our students. You know, when you mentioned that you did P22 transduction to move to define if they were recombining, that was really, really clever. And that's one of the other uses that you can give to phages. And the other one is the more popular, I think it's becoming more and more popular, is the use as therapeutics or treatments with phages. Did you mention something about that?
Yeah, so, all right. So phage therapy has a very, very long history, way before antibiotics were discovered. When people were trying to deal with infectious diseases, some people noticed that you could give bacteriophage to people, and the bacteriophage could help decrease infections. Those early studies, the problem with most of them is that they didn't have good statistics, right? So that was a real challenge. After antibiotics were discovered, people completely forgot about phage therapy in the Western world, and they shifted to using antibiotics. And the one question, the one place where it remained common was behind the Iron Curtain in the Soviet Union. So the Soviet Union was very, very poor. They didn't have access to antibiotics. And so they started using phage therapy a lot, right? One of the scientific leaders in the Soviet Union did not, he believed that if you do experiments without knowing what the result was at the end, right? That is, if you use the classical scientific method, it's not a real communist approach to science. So that shifted the way the Soviet Union did science. And so they used phage to treat a lot of people, but they never did a control, right? So phage therapy has been used on a tremendous number of people for a long, long time, but the data isn't that good. It's enticing, but not convincing. In contrast, within the last about 5 years or so, people have began to do controlled tests in humans, and the results are really impressive. In some cases, being able to save people's lives where no antibiotic therapy would work. I personally know one person that had their life saved by doing phage therapy like this. And so I expect it's going to be something that we see a lot more of in the future. But we are still in the early days of moving forward on this process. There are some phage therapies that have been approved broadly in the European Union, and in the United States, they're only approved for people who are really, there's no other treatment that can save their lives, right? But those results are really coming in, and I think it's going to change the way we think about antimicrobials in the future. And I know that some of the students in this audience are working on issues that relate to antibiotic resistance, and it completely ties very nicely to thinking about phage therapy as well as natural solutions to these problems.
Yeah, yeah. Let me show you something. One of my toys here. So small bacterial phage that I bought and I assembled. So it's really cool. So if I can get one for you, I'll mail it to you. And the other
Nice.
one, I know which story you're talking about. Is the one, the book. I think Anka, I'm not sure Rob, participated in that one. Aren't they?
Yes.
Yeah. Go ahead.
It was an amazing story because it involved people from all over the world. So it involved people from the country of Georgia in the former Soviet Union, from a university in Texas, from the United States Navy in Washington, D.C., from several groups in the San Diego region, including Anka, and a number of people from other places around the country. So it was an amazing case where a lot of people worked as a team to come up with a solution that saved a person's life.
Yeah, yeah, the book is really nicely written. There's some things that tipped me off a little bit, like the fact that the author, she is kind of esoteric and things like that, but the scientific part and the description of the case is just amazing. And I had the opportunity to meet Anka in one of these meetings in Colorado. What's the name of that meeting? I forgot it now. Anyway, my brain is not really good now. But anyway, she's really nice, and now that's an amazing story about the phages. And we have a lab practice here in our practical course, in which we ask the students to bring samples, water samples, and then we detect phages by using multiple strains: Salmonella, E. coli, Bacillus, Staphylococcus, and Klebsiella. And yeah, there's very nice and interesting stories coming from that lab practice we do here. So it's really, really cool. Phages are really cool.
Absolutely, and we have so much left to learn about them. That's a great example of where research on phages was really decreasing. It went from a very active field to a very inactive field, and it's been regenerated in part due to phage therapy, but it's a very important field that we still have a lot to learn about. And this goes for people who are working on genetic regulation and other things. Many of those important discoveries first came from phage.
Yeah. Well, let me ask if somebody else has more questions. Más preguntas, more questions, guys, comments? So,
I got another question.
Go ahead, please.
Yes, doctor, the pseudogenes can be transferred by plasmid, or are some way to induce or introduce the pseudogenes in plasmid on the Salmonella to disminute some amount of bacteria or disminute the growth of this?
So the pseudogenes are, because they're a loss of function, right? If you bring in a plasmid that carries a pseudogene and you have a wild type copy on the chromosome, then the pseudogene will be recessive, right? So this is why we had to use the bacterial virus, the P22, to actually exchange a region of the chromosome so that now there was only that single copy of the pseudogene present on the chromosome, right? In nature, plasmids move a number of things around. For example, antibiotic resistance is very commonly transferred by plasmids, but phage move a lot of genes around as well. And in nature, there are lots of phage infections that are happening all the time, and doing natural experiments like this that you'll see in the end if they provide some benefit to the organism.
Okay, thanks, doctor.
Okay, so somebody's asking by chat. It says, can Dr. Maloy explain something about the evolutionary story on Typhimurium typhi? I'm thinking when they diverted, when they became Typhimurium typhi. Thank you for the question, Marta.
Yeah, it's a very good question. So if you make predictions of when they diverged, it's really tied to when humans appeared, right? So it's after the appearance of the initial primates, but around the time that humans appeared. So this event seems to have happened, as best as you can tell with that evolutionary time scale, soon after humans became humans. What I can't answer, you could say, well, what about Neanderthals, right? I honestly can't answer that question, but I think if you think about the early human and human-like lineages, I think it's a real interesting question to think of where it came in and what we know now about the amount of genetic exchange between those ancestors that's occurred. I would think it's very likely that it potentially came about in that kind of time frame when we had Neanderthals coexisting with current human strains. But it's not a very easy experiment to do. It's really a matter of making predictions from the sequence at this point in time.
Yeah. Okay. Thank you very much. Somebody else have more questions? If not, I wanna share. Let me go ahead.
Antonio, 1 second. Let me just say for everybody who didn't hear it, I agree with you totally about the book on phage. So her name is Stephanie Stravey, and the book is called The Perfect Predator, right?
Yeah.
And it's a fun read for the science, as long as you realize that it's being written by a non-scientist. Okay, so but I think it's really a good thing to read. So it was her husband who had a real serious multiple antibiotic resistant infection. Her husband is called Tom Patterson, and I've known Tom Patterson for a long time. And so it is a great story about how a person can influence another person's health care and how something like that can really completely change science. So,
yeah.
All right, I'm sorry to interrupt, but I thought I'd just add that last plug for the book. You're on, Antonio.
No, that's true. That's true. Thank you for doing that. And by the way, whoever is interested, either you can buy it online. It's not very expensive, honestly. I bought it from Mexico through Amazon. It's like a commercial, an advertisement. And it was only like 300 pesos, which is, I mean, for a professor, some students is not that much. And there's multiple options to buy it. And I think it's a very interesting story. So anyway, I was gonna tell you that I wanna thank you by, I'm gonna email you this. So we do this to recognize your seminar, and thank you for doing this. Usually we take you for lunch with some friends, and we have nice talks, and we drink some wine or beer. But this is very unfortunately that we are not able to do that today. But I'm really, really happy that we were able to contact you, that I was able to contact you, that you were so keen and nice and kind to accept giving the seminar. And I don't lose the hope that eventually we will be able to have you here in person, and perhaps students won't be so shy to come together and then talk to you. So anyway, thank you very much, Stan. If I'm not sure you want to add something else, and besides that.
Muchas gracias, Antonio. I really enjoyed talking to everyone, and I hope someday to be able to meet all of you.
Thank you. Yeah, that would be great. There's some comments coming about that. Thank you very much for all these, and an excellent conference, and really appreciate it, Stan. It was really nice to see you again, and I look forward to seeing you again. Okay?
Right.
Thank you very
Gracias.
much. Thank you.
Hasta luego.
Hasta luego.
Hasta luego.
Para los que requieren